Hello, everyone. We'd like to welcome you to this session, The Road to Hepatitis B Cure. I'm joined by an amazing panel today, and we are excited to bring this to you. This is a unique session, I believe, and I think what we want to focus on is the journey to the cure. So not just looking at where we are with specific agents and research, but to talk about, the patient aspect. Why do we need a cure? We're going to talk about some basics of hepatitis B virology and immunology. We are going to do an overview about the approaches to hepatitis cure and how far we are. And then we're going to have Sherry Cohen talk about the affected community and what people with hepatitis B really want. And the million-dollar question, the access question, which we often don't talk about till later, which will be really actually important to start talking about now. We'll be talking about lessons that have been learned from Hep C and other drugs on global access. And then we'll also talk about the regulatory riddle and importance of research partnerships, and have a nice discussion after that. So with that, I'm going to start off with a video. This is what we're launching today as part of the session also is a series of what we're calling FAQs. So ICE-HBV is a consortium of different groups and individuals and researchers doing work on Hep B, which Harry will talk about more at the end. And we're launching the FAQs for hepatitis B. So these are basic questions. We want to bring the community up to speed with what's happening with research, as we believe it's an important partnership to have community and science walking hand in hand in this road to cure. And so we want to make sure people living with hepatitis B and the communities around them are aware of what's happening with research. So let's start with this. This is a video from Danjuma Ada, who those of you who have been in the summit with us know that he's the president of the World Hepatitis Alliance. So we'll start with that. Go ahead.
Hi, my name is Danjuma Ada. I am the president of World Hepatitis Alliance and also a person living with hepatitis B. So, just like millions of other people that are living with hepatitis B, why do we need an HBV cure and what are the benefits for patients? So first, as a person living with hepatitis B since 2004, there is no one else that needs more cure than, of course, than I do. And that is reflecting the voice of the millions of other people that are struggling and living with this virus that has a potential to end their lives. I receive emails from all over parts of Nigeria, all over Africa, and all over parts of the world of patients living in shame, people living in fear, and people that are no longer living no longer have lost hope for living, and are asking the questions, when will the cure come for hepatitis B? I need to get rid of the virus in my liver that has the potential to end my life. I need to get rid of the virus in my liver that attacks the largest organ in the body and reduces my productivity, metabolic activities, and all the functions that the liver is supposed to play. And indeed, when I got infected in 2004, knowing that I had hepatitis B was almost like the end of the road for me. I thought that was the end. I thought all my future plans, the goals of getting married, having children, will never come to reality. Why? Because I was made to believe hepatitis was a death sentence and I wasn't going to live long to live up to my ambitions. And that is why I need a cure. And people living hepatitis B need a cure to have hope for the future. Even up till now, I wake up every day touching the right side of my ribs, trying to feel if my liver was getting enlarged. I look into the mirror sometimes to see if I had started developing jaundice and checking out other symptoms. And these and many more reduces my productivity and make me to live in fear of developing liver complications too early. And I know that is the fear of many people living with hepatitis B. What if the liver fails? What if liver complications set in? What if we develop liver cancer? So then we need a cure for hepatitis B to stop the struggling, to stop shying away from disclosing, etc., to stop the fear of not living freely with relatives. Not for a husband who will fear not having intimacy with his wife because he fears transmitting the virus to his wife, or people living in a household thinking they'll transmit the virus to their to peers or to friends or to family members, or even healthcare workers who may have the fear of transmitting the virus to people whom they are working with. So we need a cure to boost the confidence and to remove stigma, to remove shame for people living hepatitis B. The benefits of HBV cure is enormous for the patient. I know the fears I've had for so many years because of hepatitis B virus infection. I know the fears I had for my wife, that why would I give my wife transmit this virus to her? But if I had this cure, why would I have all this fear? So it takes away the fear of untimely death and removes that feeling of shame and gives us confidence to live, and takes away the fear of infecting others or for even people knowing our status. So we need a cure for hepatitis B. And it is our hope that the scientific community and all the partners will invest into research and involve patients in clinical trials so that we find this cure. So we need this cure. People have been denied jobs because of hepatitis B. People have been denied visas because of hepatitis B. People have been deported because of hepatitis B. People have lost their jobs, lost relationships, lost marriages, lost a lot because of the hepatitis B condition. Indeed, we need a cure to give patients hope to live and to remove this burden of shame from the over 296 million people living with hepatitis B. And that is why hepatitis can't wait. As a patient, I can't wait to get the cure for hepatitis B. And so also the millions of people living with hepatitis B can't wait to get this cure. Thank you.
Thank you, Danjuma, for those really powerful words. As somebody living with hepatitis B as well myself, I agree with so many of those comments, and they're not often discussed or even brought up in the open. And I would say most of my patients never talk to me about these things, but they're obviously really important issues that patients wrestle with, that a cure would really be helpful for. So I forgot to introduce my co-chair, Dr. Harry Jansen. So I just want to introduce him here, and he will help us out with the session as well. So let me say, Harry, do you want to say a few words?
No, I'm just delighted to be here and for this very important meeting, because we are, as scientists, working on a cure, and hopefully we'll be successful. But it's very important to know the opinion of the patients, and also to hear their voice on how important this is to get this disease really cured around the globe. So thanks so much for inviting me here.
Great, thank you. So next I want to introduce Dr. Thomas Tu. He is actually a researcher working on the hepatitis B virus in Australia. Also a person living with hepatitis B who has launched an amazing online patient forum called hepbcommunity.org. I encourage all of you to check it out. And on his website, he's actually created a really great a community for not just patients, but also for scientists and physicians to interact with the affected community. And he's currently doing a research contest. So anybody who's here, if you have some research you'd like to share with the Hep B community, this is a great place to post it. And there's actually an award for the thread that has the most interaction. But we'll hand it over to Thomas. I believe you have a video. We'll go ahead and play that.
Hi, my name's Thomas Tu, and I'm here to explain to you the basics of hepatitis B virus infection. So first off, what is the virus? Is it alive? Is it dead? Well, it's a bit of both, actually. The analogy I like to make is that the virus is like a seed. Like a seed, it doesn't in itself grow or eat or drink, but it contains the potential for life that eats and drinks and all this sort of thing. It grows up to be something that's alive. Like a seed, the virus needs the right environment to grow, the right soil. And for hepatitis B, this soil is the liver. So when you're infected with hepatitis B, it gets into your blood and seeds into the liver, where it establishes a root system and starts to grow into a tree that produces more virus and also virus antigens that help it spread to the entire liver. So you only need a handful of these viruses to eventually infect the entire liver. And this is what happens at the start of a chronic infection. The whole liver is infected with hepatitis B, and it's producing a lot of virus into the blood, as you can see up the top here. It also produces a lot of virus antigens, such as surface antigen. And the virus itself doesn't harm the liver directly. So what it means is that there may be a large time where you don't feel any symptoms at all. However, at some stage, your immune system starts to recognize, wow, the liver is completely infected. We've got to do something about this. And the way the immune system gets rid of viruses is basically to kill those infected cells so they can't produce more virus, like so. And what this means is you have some liver damage and you lower the amount of virus DNA in the blood because you have fewer cells that are producing the virus. Eventually, the immune system may be able to control the majority of cells that infect the liver, that are infected in the liver. And what you end up with is very low levels of virus production. But the immune system, the virus is very tricky. It can produce these forms that only produce surface antigen. And the tricky thing about surface antigen is it can hide those viruses from the immune system. And so basically you can have this low-level virus still being there, but it's hidden from the immune system. So it can't get rid of it. The balance can tip the other way then in some cases, and that those low amounts of virus can infect other cells and start producing a whole lot more virus. The immune system now can see this and starts to react accordingly, as we said before, killing those virus-infected cells. And so because of this, this increased amount of virus, we see a bit more liver damage. And this can occur over and over again. And the problem with that is that liver damage builds up and we get an increased risk of liver cancer and liver disease. And that's the real issue. How can we get around this? Well, it's really important that you are monitored during your chronic infection, so we know what's happening in your liver at any one time. And we do this by doing blood tests. So these basically test the different components of the virus and the immune system, so we know what's happening and we can react accordingly. So when we get tested for virus DNA, that is the number of seeds, the surface antigen, as I told you before, the leaves, the antibodies against surface antigen. These indicate either protection or cure. E antigen basically suggests that you're in the first two phases, and anti-E basically says you're in the last two phases. So what can we do? We can treat people. with antivirals. And these antivirals are quite safe, quite effective at reducing the number of viruses that are produced. However, everything else is still there. The trees, the root system, and also the virus antigen production. So the surface antigen is still there. If you stop taking these drugs, what you do is allow those trees that are still there to produce more seeds, and the virus comes back. And that's what's the troubling thing about the current treatments at the moment. What we're doing now is developing new ways to actually treat the virus. So we have multiple routes or approaches we're trying to take. First off is just removing all the trees altogether. That would be the sort of holy grail of what we're doing as scientists and researchers. However, this is quite hard at the moment. So other researchers are also going for just suppressing the amount of virus protein production. So making it so that the leaves and the seeds are not produced anymore. And what this allows is that immune system to reawaken, to not be blinded, and be able to get rid of the virus by itself, or with a little help from us. Particularly with this third approach, we can induce or activate the immune response better and shape it in better ways to get rid of the virus. So this is what's happening at the moment, and I hope you've enjoyed this explanation of what hepatitis B biology is all about. Thanks.
Great, thanks, Thomas. So we're going to move on to Dr. Kash Agarwal. He's going to be talking about what are our approaches to hepatitis B cure and how far are we. And he'll be using the same metaphor, excellent metaphor that Dr. Tu gave. So go ahead.
Many thanks for the invite to come and talk to you today at a very important session. And thanks particularly to Sue and Harry for allowing me to speak for all of 8 minutes on what is a phenomenally complicated area that's changing rapidly. I'm happy because I'm following Thomas, and he's already given us a really nice overview and analogy that helps us all understand what we're trying to do in hepatitis B. Given the time I have, I'm going to give you my key messages first of all. First of all, hepatitis B is a complicated virus, and we're still learning a lot about it, and a lot of work is going on at this moment in time. There's still debate as to what exactly hepatitis B cure means, what the endpoint of that is, and how we're going to achieve that, and what combination, if any, of new drugs are we going to need. We have to remember we have very good suppressive drugs now, so the benchmark for our new drugs needs to be quite high. It's really important to highlight there is very significant amount of work going on at this moment in time. It's a exploding therapy area at this moment in time, but it's also fair to say we're in the early stages of getting to that mountain of curing hepatitis B. We're in the early phase of studies. And as you can see from the ESHA panel on the right, there are a lot of individuals walking up those stairs, but we're not quite sure which staircase to go up at this moment in time and where the destination is going to be. Really important to highlight that safety is critical and that this has to be a team effort, and patients are critical in everything that we do. Thomas has given us a wonderful analogy of seeds, leaves, roots, trees, planes, tractors, and fires, and I really congratulate him on that. I'm going to give you a slightly more prosaic view, and this tells us the therapeutic approaches broadly to achieving hepatitis B cure. In the first panel, you can see we can inhibit viral replication with the drugs we already have and add in new drugs. That's stopping the protein production. We can lower the viral antigen burden by RNA interference or NAPs and other new drugs. Those are the leaves. or we could boost the immune response, the host immune response, by targeting with newer immunostimulating drugs or therapeutic vaccinations. And in Thomas's analogy, that's the planes, the fires, and the tractors. This is a very complicated slide that really just hopefully demonstrates that there is a lot of new work going on with new classes of antiviral and immune stimulating drugs starting to come in to the patient arena. We have entry inhibitors, inhibitors of the core seed, the cccDNA, which is really the critical part of where we need to target. We have RNA interference and ASOs, which stop protein production. You can see immune modulators, inhibitors of hepatitis B surface antigen release, and other antivirals such as nukes and capsids and capsid inhibitors. This gives you a table of the different types of antivirals very, very broadly. Just to give you an example, there are other classes and many other companies who are very active in this field, and we really need to encourage them in continuing on with this. I highlight Hepcludex, which also may work for hepatitis delta, which is a disease that we have no therapeutic therapies for at this moment in time. And these trials are going into phase 2. Capsid or core inhibitors, which interfere with viral DNA, siRNAs, hepatitis B surface antigen inhibitors with NAPs particularly, and then newer classes of drugs. But there's a lot going on, and we're starting to see these being put together in combination. And I personally believe combination of these sorts of drugs is going to be important. Briefly, because of time, viral entry inhibitors, Hepcludex, which is an inhibitor for hepatitis B and delta, already conditionally approved for delta because we have no therapies. And we are starting to see numbers of patients being treated and seeing data of normalization, particularly in HDV RNA and ALT. And in essence, this is controlling and stopping the seed coming into new liver cells and putting down roots. Looking at capsid assembly and modifiers or core inhibitors, there are numerous of these. They seem to be safe by and large as a class. We have quite a lot of clinical experience, but whilst they're well tolerated on their own, they don't really get us to where we need to get to if we want to get to surface antigen loss. We can see resistance and there can be relapse. And these, in conjunction with nukes, may be best placed in suppressing the virus and protein production. NAPs block the secretion, the leaves that come out from cells and are coming through and seem to be of interest, and particularly a company called Replicor are moving forward with this. siRNAs and antisense oligonucleotides from companies such as Janssen, Arbutus, GSK have now got a fair clinical experience. And again, these drugs stop the protein production and therefore stop the trees coming together. What is the rationale for HBV immunotherapy? This is to boost the host immune system. These are directly acting antivirals, only target specific steps, whereas immune modulators will attack all the viral particles and boost what is an immune system that may not be completely functional in controlling and eliminating hepatitis B. But safety is going to be important, and the key will be to differentiate good versus bad flares as this clearance goes on. So activating the immune response is going to be relevant and important. This is giving you an array of the types of drugs that we're seeing, TLR agonists, be they TLR7s or 8s, and various companies are using these now in phase 1 and phase 2 studies in conjunction with antivirals. There are therapeutic vaccines, but also checkpoint inhibitors and newer unique drugs. And these seem to activate the immune response. And I remember the tractor and the fires that can be put on to try and eliminate and control the seeds, the trees, and the leaves of hepatitis B. To just finish off with therapeutic vaccines, a lot of this coming into the clinical environment now, really boosting specific responses to hepatitis B that aren't effective in patients with chronic hepatitis B. And there are a lot going through the phases, and we will see more and more around this. But again, boosting the immune system generally, the concern will be of off-target effects in other organs, or again, this flare. So we have to walk a very careful tightrope, but particularly safety is always going to be at the forefront of what we do. So in summary, I think there is a lot going on and we should be hopeful, but we should balance our expectations and we should see the roadmap ahead is not going to be easy. The first goal, and we have a good endpoint, is hepatitis B surface antigen loss, but that is not an easy endpoint, but it's an endpoint to aim for. Combinations of drugs that work in different ways are needed, and nukes are safe, and we need to have drugs that are safe together. The holy grail will be control of cccDNA, and a duration of between 24 to 48 weeks is going to be where we land, we think, but safety always is going to be at the front of our mind, and this will be a team effort. Thank you for your attention.
All right, I'll take over from here. Thanks so much, all of the speakers, basically the scientists and the doctors working on how to achieve those cure. And now we're moving a bit more to the community and the patients and how this cure would land. And we have 3 excellent speakers for that. The first one is Cherry Cohen. She is the co-founder and director of the Hepatitis B Foundation in the United States. And the title of her talk is Engaging the Affected Community, What People Living with Hepatitis B Really Want. So what is really needed from a patient perspective. Cherry is going to present live, and I give the floor to her.
Thanks so much. Let me just go over to my slideshow. Great. Thanks very much. I'm happy to be here today, and thanks for putting together this session. I'm going to talk a little bit about how we can engage people living with hepatitis B and what we hear from people around the world living with hepatitis B in terms of the impact of hepatitis B on their life and what they want. So at the Hepatitis B Foundation, we have been working on this for a number of years, collecting data and talking to people around the world in different ways. We hosted with the FDA a PFDD meeting in June of 2020. We have been working on in international surveys in a number of languages, as well as in-depth interviews and focus groups. And we've also, we do consults, patient consults, about 10,000 a year, and we worked on some data mining for the last few years on those. And what we're gathering is a picture of what the impact of living with hepatitis B is and what people want and need, both from current treatment and future treatment. So in terms of impact, we do see that there is significant physical and emotional impact among people who have hepatitis B, and it does appear to reduce quality of life and enjoyment of life. And this is considering, I think, that the paradigm, the current belief is that hepatitis B is a silent disease, and I don't think it is. When you hear from people that even if people don't have symptoms, the impact of hepatitis B is real, not only including physical symptoms, but there's a significant impact on social and emotional lives as well. Things like fatigue, pain, stigma, fear, anxiety about transmitting the virus to others, anxiety about an unknown future and progression of the disease, potentially developing liver cancer. And having hepatitis B is affecting people's decisions, major decisions that they make in their lives. Social isolation and maybe avoiding getting married or avoiding having kids, avoiding taking certain jobs because they think it may be too much of a taxing or a challenge for them, or frankly discrimination where people can't get jobs or can't go to school or can't live in a certain country. So there is significant impact of hepatitis B. When we ask about what people want from treatments, current treatments and future treatments and clinical trials, these are the overall takeaways, and this is what we hear. Above all, people want a functional cure for chronic hepatitis B. And it looks like this. So finite duration of therapy, not having to take medication for years and years and years. And when I say finite duration, when we do our surveys, focus groups, and interviews, most people ideally would like to take something less, 6 to 12 months or 12 months or less, essentially. people want to achieve and sustain undetectable levels of surface antigen. And this is particularly important internationally because it is testing positive for surface antigen where a lot of the discrimination comes from. So people who can't get jobs, who can't immigrate to certain countries or can't go to university, it's because they're testing surface antigen positive. So that's very important. People also want to achieve and sustain undetectable levels of hepatitis B DNA and have what they feel like is a reduced risk of liver cancer, liver failure, and cirrhosis. And what we hear from people is they want, ideally to have the same risk of liver cancer as someone who never had hepatitis B. And of course, we do hear about, people overall want to have improved quality of life and they don't want to feel stigmatized or be discriminated against. This gives you a little bit of a picture in a survey that we did in 2019 and 2020. We had about 1,700 people who responded, and we asked them, what's the most important thing that you could get out of a future treatment outcome? And you could see here, the three most important things were loss of surface antigen, stopping medication, and having a decreased risk of liver cancer. Stopping medication, when we delve deeper, we saw two major reasons come through. One is cost and access, especially internationally. And two is more of an emotional impact, where taking daily medication for long term was seen to be a constant reminder of having the infection, which just kind of propagated the fear and the anxiety and the isolation. So looking here, you can see also the difference between responses from US and international. In the US, most people ranked decreased risk of liver cancer as the #1 most important thing they wanted out of future treatment. In the rest of the world, internationally, loss of surface antigen was first. And again, that stems from the discrimination that people are facing. In terms of clinical trials, what we see is that that overall, there seems to be a preference for oral treatment. However, when given options of injectables and infusions, depending on the burden of life, these also seem to be acceptable by many people. In terms of burden of participation, of course, people by and large take that into play, how often they would have to go in a clinical trial, how often they would have to get checkups, whether there was a remote option, whether they could had to deal with like kids and jobs and work and side effects and safety. However, by and large, there was a very big willingness to participate or consider participating in clinical trials, 75% or more. There was some higher willingness outside the US to participate in clinical trials. And overall, as the chance of functional cure got higher, the willingness to participate in clinical trials also got higher. And then finally, we did see differences in clinical trial willingness to participate, both in regions, so depending on where people lived in the world, and also in age and gender. I want to just end with this, that people with hepatitis B deserve, I say here that people with hepatitis B need a voice at the table. I'm going to say that people with hepatitis B deserve a voice at the table and should be treated as partners in the process as we move forward in drug and clinical trial development. And I know we heard from the scientists that yes, clinical trials are happening and will be happening in the near future. And I want to challenge us to take into account the needs of the people and think about creating patient-focused clinical trials to help people around the world participate. And also want to stress the importance of clinical management of hepatitis B, because what we hear from people is that hepatitis B impacts their quality of life. And so when we talk about how we treat it and how we manage it, we can't just talk about loss of surface antigen. We also have to talk, or DNA or ALTs, normalization of liver enzymes. We have to talk about the other things. Hepatitis B impacts people physically, emotionally, and socially. How do we manage hepatitis B so that we're also taking the quality of life impact into consideration, and giving people tools and resources so that they can live better lives with hepatitis B, and hopefully one day without it. And I think that is it for me. There's just a couple of resources here. We do have, and I thank World Hepatitis Alliance for showing some of our storytelling videos throughout these few days. Anyone who's interested in sharing their story or sharing their thoughts about what they want, please feel free to email me or find us at hepb.org. Thank you.
Okay, thank you very much, Cherry, for this very nice small lecture on what the patients would want and what is important for patients. That's sometimes underestimated by us as scientists, but it's obviously very, very important. So let's move on to the next speaker, which is Jessica Burry. She's from Médecins Sans Frontières, which means Doctors Without Borders, an organization all known to you. And she will touch upon once we have drugs, whether it's the current suppressive drugs or the future curative drugs for hepatitis B, how will we be able to get global supply of that and how to deal with that? And she will use hepatitis C as an example for that, I think. So the title of her talk is The Access Question, Lessons Learned from Hepatitis C and Other Drugs on Global Access. Jessica, go ahead.
Great, thanks. Thanks for the intro and thanks for the invitation. Nice to join you all. I'm just going to quickly share my screen, which I think should be, here we go. So yeah, I learned a lot about Hep B this morning, quite helpful. And now I'm going to change a bit the track to Hep C, to look a bit at what we've done in the past few years to improve access for Hep C, and a little bit about what MSF has done, but it's part of a much bigger community who's worked on this issue in the access to medicines movement. So there are a lot of different factors that need to come together, and I'll touch briefly on various points in the different access issues that can be problematic or need to be addressed to actually get your pills into bodies. And so we'll start with the Hep C story, which probably many of you know well, where in 2013, Gilead had sofosbuvir approved by the US FDA. And it was a game changer. It was a new drug that could cure Hep C in combination with other DAAs. It meant the difference of the previous interferon-based treatments, which were fairly intolerable for patients and not very effective. And so Gilead came to the market with sofosbuvir at $1,000 per pill. So you can imagine that was well out of reach for many. But within 4 years or so at MSF, we were able to secure access to generic medicines. And at that point, we were down to about $120. And so the key is generics. It's access to generic medicines that are made in countries where it's less expensive to produce. But the other part that's important is to have multiple generics, so you have competition between them, and then you get the price to come down. So it happened pretty quickly, I think we can say, in hepatitis C, and it's in part because of the lessons learned from the HIV story from many years before. And so this is an old slide that's recycled over and over to show that, in early 2000s, we had ARVs to treat people with HIV, 10, 12, $15,000 per treatment in high-income countries, and remained well and large out of reach in low and middle-income countries. With the introduction of generics and competition between them, the prices dropped really quite quickly. And today we can treat a patient with HIV for 1 year for about $60. So in Hep C, we started treating in 2015 at MSF with the access price from the originators, from Gilead and from BMS, and we were paying about close to $2,000 per treatment. And then over the following years, we brought generics on board as they became available, and eventually we got down to $75 for 12 weeks of treatment. And so once you have generics available, that's a large hurdle to overcome, but there are many more things that also need to fall into place. You need to make sure you have a good quality of medicines, and the WHO has a pre-qualification program that addresses that. You need the generic companies to register in the countries where it's needed. And we also need to see those drugs listed in the WHO and in National Essential Medicines list, in treatment guidelines, having countries update their treatment guidelines to accommodate for the new medicines coming to market. And eventually you'll get medicines in tenders and available in national programs and in clinics and available for the people who need them. So if you want to get access to generics, then you need to consider patents. And so the patent system is the mechanism that's used generally by big pharma companies to keep their monopoly and to make sure that they're the only ones who can sell their drugs in the countries where where they maintain a patent. And in Hep C and now in HIV as well, we see that many of these originator companies are willing to give licenses to generics or to not file patents in some of the low and middle income countries. But we often see in middle income countries where it's problematic that they're considered still as lucrative markets, even though patients can't necessarily afford to treat Hep C or HIV. And so if we're looking at patents, then one of the mechanisms to overcome is patent opposition. So MSF funded some patent oppositions in the EU, in China, and India by different organizations. And this is a bit the long-term game. It takes quite a while to go through the court systems, and it's useful if there's a potential to show that a patent is not warranted, because it can also be upheld if it's considered a warranted patent. Countries also have the means in certain contexts, if it's considered as a national emergency, for example, that they can have a government use or compulsory license. And for Hep C, this was done in Malaysia to allow access to generic sofosbuvir, and again brought the prices down quite dramatically. And then we have also voluntary licenses. And so there's an organization called the Medicines Patent Pool, and they negotiate basically licenses between the big pharma companies and generics. And their licenses are always transparent. We can see, which countries are included, what different sorts of clauses can be problematic or can be useful. And we also know that some pharmaceutical companies will have those licenses directly with generics. And basically it gives a generic company a list of countries where they can sell their drugs without worrying that the pharmaceutical company has their patents there. So the patents are one thing, but actually what can also be interesting, especially for researchers, folks working at universities, developing medicines, especially in the case of Hep B, where we're still in fairly early days, is actually to negotiate provisions in contracts already. So if you're a researcher and you're discussing with a biotech or with pharmaceutical companies, other organizations, then you can already negotiate into your contracts, have access provisions so that your research will be used to get to the people who need it, and to make sure that you can have access for people in low and middle income countries, and they won't be stuck seeing these super expensive drugs available and not able to reach them. And so I would direct folks to have a look at the university's Allied for Central Medicine's website, and they do quite some interesting work looking at different universities in the UK, US, Canada, and Germany, and looking at their different policies around access. And they also help universities improve their access provisions, and with the idea to have a larger access footprint when drugs are actually coming out the other end of the pipeline. So this one Sherry touched on earlier, and I agree that civil society and community-based organizations, patient-based organizations, they're probably the most important, I would argue, for access, for research. These are the people who need the meds the most, but they're also the people who know the context in their countries the best. And so everything from civil society organizations, who are largely the groups of people who do the work in each country to get their drugs registered, to get it into guidelines, and so those folks are quite important to engage with them from the beginning, to finance the work they're doing, and to include them from the get-go in community advisory boards for research programs, etc. So last but not least is money, and this is where Hep B and Hep C are starting to come together. You would probably be quick to point out that we do have some meds that are available for Hep B. We have tenofovir, we have entecavir, we have generics of those, and they're largely unavailable in a lot of low and middle income countries. And I think it's safe to say it's because there's lack of global funding. There's no big donors who are coming to the markets, and countries and ministries of health are not making it a priority. And so we're starting to see the same for Hep C. We have, Global Fund, PEPFAR, they fund in the context of people who are also infected with HIV. Oops, and now I have their and Unitaid, funded a lot of the Hep C work that we did in the early days. But beyond that, for people who are mono-infected with Hep C or Hep B, there's very little interest. And so this is really becoming the main barrier that we're seeing in Hep C at the moment, where we do have access to those medicines who which could be quite useful for the people who are still struggling to get access, the 58 million people that still haven't been treated. So I wanted to touch on plenty of different topics, and without going too much into details for any of them, but I'll leave my contact information available for anyone who'd like to discuss in more detail. And I'll stop there. Thank you.
Okay. Thank you very much. We're moving on to the last talk, which is by Veronica Miller. Veronica has been working for many, many years on trying to get academia, regulators, patients, and other groups involved in drug development together in order to speed up drug development, and in particular also the way regulators look at that, look at trials and how to get those approved. So she's been very active, not only in the field of hepatitis B, but also in HIV and hepatitis C in the past. And the title of her talk is The Regulatory Riddle and Research Partnership. And Veronica Miller leads the Forum for Collaborative Research. Veronica, go ahead.
Thank you so much, Harry, and thank you both to Harry and Sue for your participation in the HBV Forum and your leadership and representation of the academic and also the patient concerns. So I'm going to kind of try and tie things together through a basic, very basic overview of the regulatory process. And on the path to HIV cure, the question I think everyone has, how long is the path and when are we going to get there? And just to kind of keep that analogy of actually traveling on the path, there, of course, there is the virus and there are the patients. And this is the most important traveler that we're all trying to help. But we also have the drug developers, the diagnostic developers, regulators, all the researchers, the caregivers, the healthcare systems, the payers, et cetera. And this really is an interdependent ecosystem that needs to work together for there to be progress towards an HIV cure. When we talk about the regulatory environment, the main thing really is the benefit and the risk. And regulatory authorities will always balance this. They will try to balance it. What is the benefit of the treatment and what is the potential risk? And these are so important, but sometimes it's a little bit hard to really find out exactly what the risk might be. And the regulatory authorities, there's a few key ones like the US FDA, the European Medicines Agency, which covers all of Europe, but there's so many more like the China National Medical Products Authority. In Japan, we have the PMDA, but there's so many other agencies. And as Jessica already pointed out, unless a drug is registered in country, people would not have access to it. So this is a path that all therapies will have to cross to get regulatory review and authorization. And when we talk about how regulators think, they always want to see an improvement in how a patient feels, functions, and survives. So one is we can think about that in terms of how efficacious a drug or combination treatment is and how safe is it, and that translates into benefit and risk. But this is not a static formula. It's very context dependent. Number one, the first question we ask is how serious is the disease? And we know that hepatitis B infection is a very serious disease, and also in the context of what other treatments are already available and how safe are those and how effective are those. And again, it's always a balancing act. How do we measure benefit? So normally, if we want to see how a patient feels, functions, or survives, we would do a trial that looks at people that have HBV infection and wait to see what a clinical endpoint might be. And this would take a very, very long time. So in regulatory science, what we talk about is we talk about surrogate endpoints, and these are biomarkers that if we can really find one that predicts how a patient will feel, function, and survives decades later, the path to drug development is a lot shorter. And this is where so much of the science is going on, and where we've been talking about the seeds and the leaves and so many different aspects of HPV replication. What's very important in regulatory science is how we design the trials, and this takes a lot of discussion between regulatory agencies, the drug developers, and this is also where the patient voice really comes in to inform people, as Shari Cohen has already talked to us about what are the patient needs and what is most important to them. And a lot of the discussion focuses on how do we know that we have achieved a cure or a functional cure or a partial functional cure? There's so many different ways of describing it, but we basically want to know if we take patients off of treatment, will the virus stay suppressed afterwards? And we all agree that we need combination treatments to achieve that. But every time you put two drugs together, you have twice the chance as well for safety issues to come up. And so how we develop these clinical trial designs is of great importance. The road can be rocky. And on the road that we're all traveling on, we do have signs, and we see speed bumps and hazards coming up. When there's a first-in-class drug, I refer to that as trailblazing. And very often, the first drugs in a new class are the ones where we really learn a lot from and then can approve the next generation of drugs in that class based on what we have learned. But we do see anticipated, but also unanticipated toxicities. And one of the major ones here is what we call drug-induced liver injury. And this is especially serious when there's already an underlying liver disease, such as hepatitis, or also, for example, fatty liver disease. And there will be drug-drug interactions. And again, because so many drugs get metabolized in the liver, if you have an underlying liver disease, that actually may make things worse as well. So this is why on the whole, regulatory authorities are very cautious. We want to make sure that that we eventually can cure patients or at least keep the virus suppressed for a very long time, but we want them to live a healthy life. And we have seen many times already that drug-induced liver injuries and other toxicities can really stop a program dead cold. And so that means then that any other combination programs this particular compound was in with other pharmaceutical companies, all of that gets stopped. And when you combine two different drugs, it's so important to be able to say where the toxicity is coming from, and so that we can safely continue the trials. But of course, we do want to all work together, and on the path, we have many opportunities to come together, and especially when we talk about combination therapy. And there are many research partnerships amongst academic networks. It's really wonderful to see the international collaboration that is really going on amongst universities, but also how the patient organizations and networks have really started feeding into this. This is so important. And we saw that today in these presentations. Industry are also collaborating. It's not just the drug developers and the biotechs, it's also the diagnostic companies that need to be involved. And then, of course, we have many international organizations, such as the Forum for Collaborative Research, which has the HPV Forum. ICE HPV is another very, very productive international organizations. We have the Hepatitis B Foundation. We have all of these groups that are really helping to build spaces such as this one, where the fellow travelers can come together with the patients that carry the virus. And just to finish off, again, just the most important aspect is for drug development and regulatory approval, we always have to look at the benefit and the risk, and we have to pay very, very special attention to the risk. And with that, I will stop. Thank you.
All right. Thank you very much, Veronica. I hope that all of the speakers can turn their camera on so that we have you live for the Q&A part. I hope Sue is still there. Is she?
I'm here. Thanks.
Yeah, once more, thank you very much, Veronica, for bringing industry, regulators, academia, patients together over the years and doing such a wonderful job. There are a few questions in the chat. Maybe, Sue, do you want to go ahead and ask the first questions to the speakers?
Sure. We had some already answered in the Q&A, so those of you who are on the platform, feel free to look at those. But let me ask some of the ones that probably will be better in discussion since they're not really quick answers. But the first one goes to Sherry Cohen. Says, thanks, Sherry, for a great presentation. Do you think that current clinical trial designs are taking these patient concerns sufficiently?
That's a great question. The word sufficiently is what gets me. I do think clinical trial developers and drug developers are starting to think about patient-reported outcomes and patient-centered, patient-focused clinical trials. There actually is quite a lot of discussion on that now, and I think the will is there to do it. I think we still have a lot of work to be done, but I do think it's happening. I think one thing is COVID-19. I think because things changed for clinical trials during the first lockdown, where we learned that with clinical trials, maybe you can do some things some visits via telemedicine. Maybe you can have visiting nurses take blood and do so you don't have a person traveling to a site every week, for example. I also think we're learning from other diseases and clinical trials where maybe there are community forums and apps and other kinds of social support that we can include for people who are going through the clinical trial process to make it easier for them. And so I do think it's evolving in a good way. I also want to say that the Hepatitis B Foundation, in July, we're going to announce 2 new global community advisory boards, one on hepatitis B and one on hepatitis D, with representatives from around the world. And the goal of this community advisory board is to have a group of people living with Hep B and D who can reach out to clinical trial developers and actually say, hey, come talk to us about this. We can help you develop patient-focused clinical trials. So hopefully we'll get even better in the future.
All right, thank you. A lot of questions have been answered already in the chat. I have a few questions myself as well, if that's fine, Sue. So Jessica, thanks so much for your talk. I fully agree with you that we should get the drugs out once they're developed and licensed as quickly as possible for low cost. But I work also on the development side, and companies, people typically see the drugs that make it to the market and then start to complain how expensive they are. But a lot of drugs fail in the pipeline, which costs the companies hundreds of millions, if not billions of dollars, right? So there is a balance towards development of new drugs, which they can only do when they have sufficient money, basically, and how to get the drugs to a reasonable price to particularly low-income countries. Can you maybe specify on that balance and how we should act within the hepatitis B arena?
Yeah, sure. So yeah, it's that's a common argument that we hear, but I think so far we haven't been able to see actually what is spent by pharmaceutical companies on the actual R&D. They're not too keen to open a book and tell us what the development of a drug costs, even if we want to take into account the development of the many molecules that might not ever come to market. And so I think what we want to avoid is that, you know, we're paying twice, where, you know, public money is being used to finance research and then again to pay for the drugs later, especially when they can be quite expensive coming out. And we know in the example of Hep C that phosphorus, the cost of manufacturing was quite low. There were some studies done at the University of Liverpool on the cost of production, and they showed quite early on that based on the cost of the pharmaceutical ingredient, the cost to produce it was actually about between $50 and $100 for a treatment. And so we could see that with an $84,000 price tag that there was something wasn't quite lined up. And in that actual example, Gilead had there's a U.S. Senate investigation that showed actually how they priced the drug, and it was related to the model of insurance payer in the U.S. And they didn't look at the cost of research nor the cost of producing the drug, but it was related to, in fact, what the insurance companies were willing to pay. So I think if we look at, what
I'm not talking about the cost of the drug. I'm talking about the cost of all of the drugs that don't make it. And I'm involved in that research, and I see it happening also in hepatitis B, that many drugs at a very late stage after very expensive trials have been done don't make it, right? So I agree with you that there should be more transparency maybe on the side of the companies on what they spend there, which is significant. And hopefully we can find a middle ground there, I would say, because it is a bit of a balance looking at, and I mean, I'm not part of any company, I'm just an academic doing research and trying to help them out. But I see a lot of drugs fail, and then at one time one comes up, and again, that they need to have some sort of return of investment, which should not be ridiculously high. I agree with that.
Yeah, it's a hard balance to find, but when we look at the profits they turn over and that we see in the shareholder meetings every year versus the access and availability of the drugs, then I think we're far from the balance, so we still have some work to do.
Yeah. I had also a question for Thomas. Thomas, can you tell me when do we expect the new drugs which would have the potency to cure people with hepatitis B on the market?
On the market? Well, I don't know on the market. I'm just an academic scientist like yourself. So again, that time is unknown to me. What I see in the field is maybe not something that's absolute, maybe just like we saw in hepatitis C, in some years that there may be a compound that allows functional cure in only a fraction of patients, and it's up to us to sort of define that fraction of patients who are more easily treatable or something like that. That is my prediction, I suppose, and we slowly build from that, I guess. Those are really my thoughts, but nothing. I wouldn't put money on it.
Well, maybe I can say something. It won't be in the coming 3 years, 5 years, right? So
right.
Phase 3. So it's a difficult road. I think we have been very successful in the hepatitis arena with curing hepatitis C. Companies are reinvesting their research dollars now in hepatitis B, which is a less easy to cure disease, and it will take a couple of years to get that done. Hopefully, it will be a long and winding road, right? Yeah.
Agreed. Yeah. So I agree.
Yeah. Okay.
There are a number of questions, which I'm going to kind of put them all together, but there are obviously a lot of questions about access and once we do have the drugs, how we're going to get it into low, middle income countries. And, kind of learning from Hep C, I try to figure out, what can we do ahead of time now, right? Maybe this goes to Jessica. I know you kind of laid out all the number of agencies and things like that. I think this is very helpful for people to see that it's not so easy to just go into, global access. It really is done by regions and countries, and there's quite a lot of steps. So it'd be helpful, I guess, what you think we can do now as we're preparing. We don't have anything right now, but what are the things, either the community or researchers or people who care can do to kind of prepare us? And I would say in the U.S. we've had discussions like, insurers need to be part of the conversation. So what we found out with Hep C is they were not aware that these things were coming out so quickly, and once it came out, what they ended up doing is putting a lot of blocks in, right? So there's a lot of pre-authorization, which ended up slowing down access for a lot of people. So we've had that discussion. There's another quick question about if there's any way to speed up drugs becoming generic. And I think that may be different in different countries as well. But I'll let you answer, Jessica.
Yeah, sure. So I think a lot of that work can be done before we actually get an approval. So already, even for us, we're following the pipeline. So, if there's something interesting that looks like it really could be a game changer in Hep B, then ideally, different organizations will come together to start looking at, are there going to be patents that are blocked, and are they going to need patent oppositions, or is this company already discussing with the Medicines Patent Pool to actually make those agreements with generics in advance? And then to have the generic start the development early on. And we saw a really nice example of that one in pediatric dolutegravir, where when the drug was from the originator was approved by the US FDA, the generics were already, pretty much had it ready to go. I think they just needed to finalize the bioequivalence, and then it was approved, the generic formulation, a few months after. And so that was really key examples to highlight all the people that basically came together, and it was quite well organized, I think, to have the generic companies on board and start the development early. But it means engaging with them and to make sure that they're aware that these drugs are coming, that they can be important. I mean, it's a huge population of people living with Hep B in various corners of the world. And so I think it will be an interesting market for generics to come into. But it is a matter of keeping an eye on the pipeline, what's interesting, and engaging early, and that means taking risk. That means potential, like Harry was saying earlier, that some of these drugs might not actually make it, and so there's potential that generics will do the investment to develop, and it might never work out. And then it's also to engaging with the regulators, as Veronica was discussing about, to make sure if it's a game-changing molecule, that perhaps they can have an expedited review, or through the WHO program, they have a mechanism to fast-track registration in low- and middle-income countries for the participating ones. So there are quite a few different things that are useful and that have developed over the years to expedite access to generics as quickly as we can. It's just a matter of aligning everybody on the same page.
Great. Thanks, Jessica. So Harry, I think we maybe only have time for another question and then for your closing remarks.
Yeah, maybe I have a question for Veronica as well, because you have a lot of experience in licensing drugs, etc. And also in other infectious diseases. What would you see as the major hurdle for hepatitis B new drugs get approved, Veronica? I think you're muted, Veronica.
It depends a little bit on what you want to approve it for, right? If you want another virologically suppressive drug that's better than tenofovir, for example, that would be relatively simple because we know how to do that. If we want to approve a drug for finite treatment, that's really a hurdle because we have to define and figure out how do we measure that finite treatment, which means taking people off of treatment to see if we take them off of treatment, do they really continue to be virologically suppressed? And that just carries so many other risks and nuances with it. We just had a wonderful discussion last week between the American and European societies on how do we really measure that, and what are the risks of viral rebound and flares, et cetera, in liver flares in that situation. So that's a little bit more complicated. So I see the real hurdle is really understanding what it is we need to measure and how we measure that in a way. And to answer Jessica, yes, there are many paths for accelerated approval there's so many designations the FDA and also EMA has for breakthrough designations, fast tracking, et cetera. So that's all there. And in general, I have found that the regulatory agencies really come at this from a public health perspective. But, we often think they're very conservative, but they're really conservative on the side of safety, and they should be. The other major issue, I think, is that some of the safety signals might not really be detectable until much later, once the drugs are actually on the market. So the pharmacovigilance program will also be very, very important. And finally, just one more comment on the issue of generics. I think small molecules, are, the generic process in terms of how to synthesize these chemical entities is one thing. But we also have a more complex kind of therapeutics that we're developing for hepatitis B. And I think the generic scenario in that case will have to really work together to figure out how we can increase the access to some of these compounds that you just don't kind of, easily synthesize, like, for example, sofosbuvir, which is easy to manufacture.
Okay. Thank you very much, Veronica. Thank you very much for all the speakers. And I think having been in this area of hepatitis research for, let's say, 30 years, we're kind of privileged that we've done a lot of work over the years. When I did my PhD studies in the 1990s, we had nothing for hepatitis B, we had nothing for hepatitis C. Hepatitis C is now a fully curable disease, and hepatitis B is a fully suppressive disease, and we're trying to make that last step to get hepatitis B cured as well. And it's pretty unique, not only in liver disease, but in medicine in general, to have two of these diseases recently discovered and being treated so well currently. So I think we've done a great job. The gist of this and the next challenge is how to get hepatitis B cured, and particularly how to serve all the patients that are so much underserved around the globe and don't have access to treatment. So with that, I would like to close the session. I think there's two more slides that I would like to present. Again, thanks so much, Sue, Wayne, for organizing, and all of the speakers. Were phenomenal talks. I'd like to remind you that this was organized on behalf of ICE-HBV. It's a collection of leading HBV researchers dedicating really to advance science to get hepatitis B cured and affordable around the globe. So there is a specific survey you can fill out. You can contact them through the links that you are seeing here on the slide. And I would also like to tell you and advocate to spread the word about HBV cure to create more awareness and advocacy, because we really need more input also from, I would say, ministries around the world to help us out. There is an ICE-HBV virtual booth that you can visit. And I'd like to thank them once more. And I can have the next and last slide. So the acknowledgements goes in particularly to the governing board and to the management team of ICE-HBV. So on behalf of everyone, I thank you for joining today. I wish you a very pleasant morning, afternoon, evening, wherever you are in the globe. Thank you very much.
So we have a closing video, actually. This is part of the FAQs that ICE-HBV is launching at the World Hepatitis Summit. So we're going to close with a video from Thomas again, because he does such a great job explaining these complex topics in an easy to understand way. And this one is about understanding the difference between what does cure mean and looking at sterilizing versus functional cure. So go ahead and play that, and thank you all.
Hi, my name's Dr. Thomas Tu, and I'm a scientist specializing in hepatitis B research. Thousands of scientists all over the world like me are working on a cure for hepatitis B. But what does a cure really mean? You might think that this is a simple question. It's just getting rid of the virus. But because of the complex nature of the virus, there are actually 4 levels of cure, each building on top of the last. Having these different levels helps guide our research to scale this incredibly difficult mountain of a problem. So here's the virus, and it infects your liver cells. The virus sets up a factory inside. It starts making proteins like hepatitis B surface antigen, and makes more copies of itself, more viruses. In some cases, the virus makes defective factories that can only produce surface antigen. When you take antivirals like entecavir or tenofovir, you shut down the virus-making part of the factory, so Hep B DNA levels go down. Everything else is still working. A level 1 cure, known as a partial cure, is simply getting a person to this state, but without having to take a pill every day. This has the benefit of saving you money in the long term because you only need to take it for a short term. for a short period. It limits the risk of transmitting it to others and slows down or stops liver damage. I mean, this is pretty good, but we're just getting started. A level 2 cure, also known as a functional cure, is level 1 plus the loss of surface antigen. This is the equivalent of your immune system naturally clearing the virus infection. A level 2 cure is also linked to better disease outcomes and also helps with clearing a lot of the stigma and discrimination associated with Hep B, because so many insurance, job and visa checks are linked to surface antigen in our current systems. A level 3 cure, a complete cure, is level 2 plus destroying all of these factories. Basically, this means there's no risk of virus reactivation, which we know can happen if people with a level 2 cure are heavily immune suppressed. This provides a lot of peace of mind. This cure is extremely difficult. It involves going from 10 million factories down to 0. If you miss one, it can come back. Now, finally, the pinnacle, a level 4 cure, a sterilizing cure. This is level 3, plus getting rid of these DNA fragments that are buried in our DNA. We know that there's a link to these fragments to liver cancer, but by how much we don't know. Basically, if we get rid of these forms, it's like we have never been exposed to the virus at all. And we think that this kind of cure would completely eliminate any additional risk of liver disease or liver cancer. So I hope that helps you understand what we mean when we say Hep B cure. I'm off to get working on this mountain, but to learn more, please ask questions in the comments, look at other videos by ICE HPV, or join for free hepbcommunity.org to chat with our experts and network with other people with Hep B. Thanks for watching.